So Jill has entered into very extensive network analysis, as was mentioned by Carrie. These people are not part of a formal seed system. They get new seeds from their friends and neighbors. And so she's done very massive surveying. This is only a very, very small piece of her data set, where she's looked at how people exchange information and share bean seeds. And you can see, looking at these 4 families in one village as an example, the husbands' networks are more extensive because the men they don't really work on the farm. They trade, they drink beer with their friends, and so they can, if Penn State comes up with a new seed that helps them buy more beer and marry more women, that information is going to be spread very rapidly through the community. The women are stuck close to the home because they're taking care of the children, they're taking care of the crops, and so their networks are not very extensive. So this presents a challenge because the seeds that are preferred by women are not going to propagate through the system. And this is something we're just dealing with this past season, because now we have Celestina's new varieties. So working with our Mozambican colleagues, Jill has developed systems to flood these markets with small seed packets, kind of what Carrie talked about, to kind of circumvent all these informal networks. Everybody has access to the new seed. Everybody can try it out. And we'll see how that works out, but it seems to be working out well so far. This technology was also, through funding from the McKnight Foundation, applied in China with soybean. This is work done in collaboration with my former student and friend, Xiaolong Yan, South China Agricultural University, who tragically passed away from cancer several years ago. Using phenotypic selection for shallow-rooted traits, people in his team have gone from the plant on the left, which is what soybeans look like when we started after several thousand years of cultivation, to the right, which is what we think they should look like. And this has been very successful. 15 new phosphorus-efficient lines have been released in South China from Xiaolong's program in provinces inhabited by 300 million people. So we've also seen the same kind of patterns and traits in maize for phosphorus efficiency. It's not something that's specific just to legumes. But with maize, phosphorus is important, but water and nitrogen are even more important. In poor nations, water and nitrogen are primary limitations to yield. In rich nations, all over the earth, drought is the biggest risk to crop production. And nitrogen fertilizer, where fertilizer is used, is actually the primary energy cost, the primary economic cost, and the primary environmental cost of producing corn. And the cost of fertilizer has risen dramatically along with energy costs. So it's surprising that over half of applied nitrogen fertilizer to corn is never even taken up by the crop. It's wasted, it goes right into the environment, causes pollution. So we saw this as an opportunity to do something about improving nitrogen acquisition. This is what happened in the spring in Pennsylvania. This is a simulation, very realistic simulation of a nitrate pulse moving down into the soil as a corn plant grows in the field in Pennsylvania. Every time it rains, it pushes the nitrate down into the soil. It's a race that the roots try to catch this before it's lost and goes into the environment. This is why we lose a lot of nitrogen. So our idiotype for nitrogen capture and for deep water capture is steep, cheap, and deep. Steep being architectural traits that position the roots deep in the soil quickly. Cheap being traits that reduce the metabolic cost of soil exploration. I'll talk about that in a minute. And deep is where you want to end up. Now, this is a hypothetical idiotype. Everything on this diagram is being researched in my lab right now. Most of it is unproven. I just want to give you one example today of one of these traits, and that is this high RCA, high root cortical parenchyma. Root cortical parenchyma are air spaces that form through programmed cell death in the root cortex. There's natural genetic variation for this trait. These two plants here are rills from the same population. The one on the left has an intact cortex. The one on the right has formed parenchyma from these cortical cells. Some years ago, we hypothesized that it was known that phosphorus and nitrogen stress increase parenchyma formation, and we hypothesized that this is because it made the roots cheaper. By converting living cells to airspace, you make the roots less expensive, and our analysis was that would help the roots grow through the soil and capture resources. That was the idea. In fact, this shows that as you increase parenchyma formation in maize roots, you can cut the respiration of those root segments in half. It's quite a significant benefit. This is a map of parenchyma formation in maize root. It's developmentally regulated. It's expressed differently in different organs, so it's a complex spatiotemporal map. So we're using modeling to look at this. So you can see how it's forming in the root system here at 42 days after planting. The hotter colors indicate more parenchyma. And since we know how parenchyma affects respiration and nutrient uptake and so on, we can use these kinds of models to estimate whether this benefit is likely to be important, whether the small effect from more Arenchyma production was really going to be beneficial. So here we have a simulation of a maize plant with and without Arenchyma growing through a low phosphorus soil. So the one with Arenchyma has a little bit less phosphorus and respiration in every root segment, so it's able to grow a little bit more. There's a shoot model here that you're not visualizing. That more phosphorus results in more photosynthesis, which results in more carbon and more root growth. So it's an autocatalytic virtual cycle. And after the end of the 42-day simulation, you see the plant with Arenchyma actually had a bigger root system, bigger growth, according to the model. In fact, the model, and this is part of the PhD research of Johannes Postma, showed that as we increase the stress, as we go from 100% growth on the right of the x-axis to the left, as we increase the stress, the benefit from Arenchyma under nitrogen stress increases up to about a 60% increase in growth under nitrogen stress. For phosphorus, once again, up to about 50-60% increase in growth because of Arenchyma. For potassium, even up to a 70% increase in growth. Now, admittedly, I did not believe these results when Johannes showed me them. I thought this is too positive. I couldn't believe that Arenchyma had such a big effect. Of course, the modeling just kind of gives us ideas for our field work. So this had to be confirmed in the field. The model also suggested that even at high fertility, Arenchyma could reduce your need for nutrients. For nitrogen, a 13% reduction in nitrogen requirement, which in terms of U.S. agriculture would represent $350 million a year for U.S. corn growers and significant benefit to the environment. So this is work of Patampong Sanguilai in the field. This is our field site in South Africa trying to confirm these modeling results. So he's growing high and low Arenchyma maize lines that have the same genetic background. In the greenhouse and in the field, those high Arenchyma lines shown in blue here under low nitrogen were significantly had deeper roots, as the modeling suggested. High Arenchyma increased growth in low nitrogen soil. That's the African field site in the background there. In South Africa, we had a 60% increase in growth under low nitrogen by Arenchyma. That's exactly what the model predicted. In greenhouse mesocosms, a 60% increase in growth. So at this point, I was apologizing to Johannes for telling him I didn't believe his model. And in a less stressful environment in Pennsylvania, a 33% increase in growth under low nitrogen. In fact, in Pennsylvania, we had a 60% increase in yield under low nitrogen associated with Arenchyma. Now, another deeper source is water, so we thought Arenchyma would be useful in drought. This is work of Jinming Zhu using rainout shelters in Pennsylvania. So Jinming showed using high versus low Arenchyma lines, and now the water stress here is red and the well-watered is blue. The high Arenchyma lines had significantly deeper roots under water stress. They had better midday water status under water stress, and they had 8 times better yield under water stress in the field than the high Arenchyma lines. This is what those ears look like. Those water stress is the top panel. You can see the lines with low Arenchyma look pretty bad. Those lines with high Arenchyma look pretty good considering there was no water given these plants. 3 weeks after planting, we entirely shut off the water supply. So once we got to thinking about cheaper roots, a number of traits come to mind. This is one of the ones we've been having good results with. This is research of Joseph Chimungu. And this is natural genetic variation for the number of cortical cells across the cortex in maize roots. And here you see one genotype has 6 cortical cell files, the other has 13. In fact, there's 300% variation for cortical cell file number in maize lines. And just as with Arenchyma, that is directly related to root respiration. We can cut respiration in half by reducing the number of cortical cell files. And by cutting respiration in half, we can increase the rooting depth of these plants under drought. And that's associated with better plant water status under drought. And that's associated with better growth under drought. And that's actually associated with better yield under drought in Africa and in Pennsylvania. So we're deploying this technology in collaboration with colleagues at Luana in Malawi. This is maize breeder Moses Maliru working with a farmer evaluating these new maize lines. As was pointed out by the previous speaker, it takes a lot of years to go from an idea to field impact, but we're starting this process, and Moses is having very good results with this trait and other traits for cheap maize roots in the field in Malawi. Let me talk a moment about phenotyping, because whenever you start talking to breeders about root traits, they're very concerned about how they're going to measure them, and they're going to get all dirty, and you're going to dig up the plants and stuff like that. So we've got a technique called shovelomics that all you need is a shovel and a graduate student. This is James Burge. Hopefully an inexperienced graduate student doesn't know what they're getting into. 2 minutes per plot. We're currently going through about 10,000 lines per year of various crops, and basically you're just digging up roots and looking at them. And you can see here at one of our field sites in Africa, we're digging up a bunch of maize plants. Here's postdoc Eric Nord in the foreground, visually scoring them against scoreboards that we've set up. In the background, some technicians photographing them. Working with Joshua Weiss's group at Georgia Tech, we developed a software called DIRT, digging into root traits, that takes that image and then extracts a bunch of interesting parameters from that photographic image. Now, next month in Germany, we're having a workshop to actually automate this. So the idea is you drive a tractor through the field, and the tractor is digging up your root system, slicing it, rinsing it, imaging it, and you're getting this at a rate of about 3 miles per hour as you drive through the field. And then we're going to have another tractor implement behind that one with a graduate student writing up the paper. This is an example of what variation we see just from this very simple technique. This is angle of brace roots. You can see that the histogram is 5,000 genotypes from the nested association mapping panel. You can see the variation is huge. The theme of the workshop is variation, huge genetic variation for everything below ground. We go from 10 degrees from horizontal to 90 degrees. So basically from vertical to horizontal roots. That's what we see. And now Hannah Schneider's doing genome-wide association mapping of this trait from 8 replicates of 1,500 entries over 4 years. That's 12,000 plots of analysis. And you can see we have some loci on chromosome 1 and 2 that look quite interesting for this particular trait. For anatomical traits, we had to develop we were doing hand sectioning, which doesn't work when you're working with geneticists. They're always thinking of thousands of more lines you want to be phenotyping for them. So we developed this technique called laser ablation tomography, where we're basically just disintegrating a plant tissue using pulsed ultraviolet laser and photographing the tissue as it's being destroyed. This is what the system actually looks like. Used military equipment. It cost about $35,000 on the used military market. There's a lot of good used military equipment out there, thanks to our defense budget. This would have cost like $150,000 had we bought it new. So this is what it looks like real time. You're looking at a maize root that's going into a sheet of laser light and it's being obliterated, and we're just photographing it. It's a real-time image. So then the root is gone, but now you have a three-dimensional reconstruction of that tissue. And this works with woody tissue, stems, leaves, cell phones, whatever you want to do. We're zipping into a xylem vessel here. And you can see this is actually a low-resolution image that just took a couple of minutes to generate. But then we're pulling out in the yellow. You can see there's a Renkema lacuna, so we can quantify these volumes in three-dimensional space and do anatomical analysis in a high-throughput way. So once again, we have two-dimensional image analysis software we developed called RootScan. We're working on a three-dimensional version. But RootScan will pull out 15 or 20 traits from these sections, and then we can do genome-wide association mapping. This is more work of Patsapong Sengwalai. You can see for root cortical of Renkema, we have a number of interesting hits that deserve further investigation. So I was talking earlier about the connection between anatomical and architectural traits and trait interactions is something we have to understand better. So in that regard, we developed this new model. Here's the SymRoot model at 42 days showing you a Renkema as a heat map. Now, Jia Wu developed a new model called Root Slice now that's allowing us to go into anatomical traits. So this is an explicit functional structural model of anatomical and tissue traits in maize. So now we can add, even subcellular traits are shown here. You can see the vacuolar volume there. So basically we can look at the interaction between anatomical and architectural traits in silico, and we need to do that because in my lab we're looking at about 20 different traits right now, and the possible combinations of those, even if they only exist in 2 states like long, short, shallow, deep, present, absent, is well over 1 million phenotypes. There's no way we can do field work with all those things. So the modeling helps us kind of determine where we should focus our efforts. And this shows you an example. Here we have root cortical parenchyma on one axis, number of crown roots in maize on another axis, and then a high and low nitrogen environment. And without going into that graph, I just show you that there's an interaction between an anatomical trait and an architectural trait and in the environment, high and low nitrogen. So we're hoping that the modeling will allow us to explore the millions of possible phenotype scenarios and focus on those that are most intriguing before we do field work. Let me take a moment to talk about translational research. This is what I'm interested in my work, the agronome, where you're interested in productivity and sustainability and equity and those kinds of issues. Most of the folks at this meeting are working at this level, of course, at the genome level, and this is the basis for the variation we use for breeding. There are some people who think we can go from genome to agronome with brute force methods like genomic selection. I think they're wrong. I think that we really need to understand the phenome and the traits and the trade-offs and how they interact to really make sense out of this. It's very complex. The phenome is at least as complicated as the genome. It's more important than the genome. Natural selection acts on the phenome, not the genome. Breeding acts on the phenome, not the genome, or at least traditionally. You select your husbands and wives and girlfriends and boyfriends on their phenome, not their genome, hopefully. It's interesting how we focus so much on the genome, which is really cool, and it's amazing what we've accomplished, but we've kind of neglected the phenome. I think we really have to take a better look at this, and I think this is, the young people here, I urge you to think about the phenome as you go about your work. It's going to be where we need to work in the future. There's a lot of unanswered questions we need to address. So let me summarize what I was talking about today. Root architecture is an important component of soil resource acquisition. Topsoil foraging is important for phosphorus acquisition. Subsoil foraging enhances nitrogen and water acquisition. Several anatomical and architectural themes affect the metabolic cost of soil exploration, which is an important component of foraging efficiency. Bean and soybean genotypes incorporating root genes for superior phosphorus acquisition are being deployed in Africa, Asia, and Latin America today. The steep, cheap, and deep idiotype for water and nitrogen acquisition appears promising. High-throughput phenotyping platforms are available, and numerous genetic leads have been identified. Prospects for the second green revolution. Breeding for yield at low fertility has been successful in the very few cases it has been attempted. Aluminum tolerance is a technology that is being deployed today in Brazil. Phosphorus is more complex, as I'm showing you today, but it's working. Nitrogen is quite complex, but is promising. Manganese tolerance is being deployed today. I don't know of anybody on Earth who's looking at potassium, calcium, magnesium efficiency, but they should be feasible. Recent advances in root biology are making it possible to design root systems for specific environments. The bottleneck, in my opinion, and what I hear from breeders in the private and public sector is not genomics, it's phenomics, is what traits do we want. I've had people from Monsanto tell me they could make a corn plant get up and dance, but they just didn't know what it was supposed to do to be drought tolerant. We need more information about long-term system impacts of this technology, but it should be benign. There's no resistance breakdown to abiotic stress tolerance. It's not scale positive, meaning you're not benefiting just rich people like the Green Revolution did. You're benefiting both rich and poor farmers, and it reduces pollution in rich nations. Certainly, as soils degrade, fertilizers become more costly, the population expands, the need for this will only grow. I've been summarizing the work of many people. Thank you very much.
So we have time for questions. I'm going to look over here because apparently I wasn't watching over the microphone over there, but there's nobody there. So.
Hi, Jonathan. I was curious, it seemed that different architectural features are important. Different architectural features are important for different environments. Are there architectural features that might do well in every environment, or do we really have to look at the local situation?
Yeah, that's a very good question. And, we see much more phenotypic variation below ground than above ground, as you know. And I think that's telling us that there's not just one ideal solution for below-ground environments, that plants have developed multiple solutions that co-optimize multiple constraints across a range of soil environments. And so, I think of this myself as simplistically, there's high-input environments like the U.S., and there's low-input environments like Africa. But even within those low-input environments, there's acid subsoils, there's low phosphorus, there's manganese. I mean, there's things eating roots. There's intercropping systems. So I really think that we need to start thinking, and this is one of the challenges, is thinking about which traits work best in which environments. Even these cheap traits, it's like, why is there natural genetic variation for traits that make the roots cheaper? There must be trade-offs. If you don't have, for example, with Arenchyma, there's no cortical cells, so there's no place for mycorrhizas to live. It might be easier for pathogens to get traction. There's reduced water and nutrient transport. So I guess the short answer is no. I don't think there's one root system that's good for everything. And that's one of the challenges to the field.
Jaswinder Singh from McGill University. Quick question. First of all, thank you very much for a very nice talk. Basically, I also agree with your phenome stuff, but is it possible that the root architecture which we are seeing by digging, can we relate with a marker, for example, which we are just looking at leaf level?
You mean a phenotypic marker or a genetic marker?
Genetic marker.
Yes, absolutely. I mean, as much fun as digging roots is, I'm expecting most people don't want to do it. I encourage you to do it if you haven't looked at roots. Even Arabidopsis has cool roots. But a lot of breeders don't want to do this. And so the idea is, once we figure out the genetic control of a trait, and then breeders, especially in the countries that have access to molecular breeding resources, they can use the markers rather than looking at the roots. So that's why we're doing the genome-wide mapping and these kinds of things. But a lot of the programs we work with in Africa, they don't have tools for molecular breeding. They don't, like Celestina doesn't even have reliable electrical supply. So for her, she's just looking at phenotypic selection, and that works for her. But if you have access to molecular breeding tools, then the markers should work.
Thank you.
Yeah, great talk. I was wondering, of the high RCA roots, do they also enhance submergence or waterlogging tolerance?
They do, and of course, 90% or more of the literature on Arenchyma relates to flooding tolerance, because those air spaces allow the roots to respire under hypoxic conditions. So Arenchyma helps you under drought, according to our data. It helps you with low nitrogen, according to our data. It helps you with low phosphorus, flooding. So this is the question that we were talking about with Jose. Is like, well, why don't all plants have Arenchyma all the time? And that's one of the phenome questions we'd like to answer before. You have to be careful when you're working with breeders, because it takes them a long time to develop a line, and then you don't want the farmers to pay the price if you haven't thought about your technology enough. So that's why we do field work. After several years of field work, given all the costs and benefits and trade-offs, if there's still a benefit, we'll go ahead with it. But I think that's a good question. Why, if it's so beneficial?
What do you think the trade-off is for the steep, whatever the steep,
steep, cheap, and deep? Yeah.
You asked the question, like, why don't all roots have this? Do you have an idea of what the trade-off is for plants? Why haven't they developed this already, or why haven't they been selected in breeding programs?
Yes, well, I think that's a good question, and breeders have unconsciously selected for plants that do well in their environments. Traditional people have done the same thing, but as I was talking about a moment ago, there's a lot of different soils, and there's different types of environments, and we have to begin to looking at that root phenome and unpacking it and understanding it if we're going to really make intelligent progress and not just rely on this kind of subconscious selection. Thank you.
Let's thank Jonathan for a wonderful talk.